So today's topic is lens anatomy and metabolism of lens. So we go through quickly. The lens is, as we know, it is, I think, bread and butter of the world. Bread and butter of the world. Many of the surgeries in the world are done on the lens only. Around, I think, 80-90% of the surgeries in the world are done on lens only. So everybody should be well versed in lens anatomy, surgical or medical, understand the metabolism of the lens. So we go first of all, what is the uniqueness of the human lens? What's unique about it? So, first of all, it's a really highly organized system in the human eye. So, it has some functions. First of all, what's the function of the lens? First of all, the function of the lens is a refractive medium. It's a refractive medium. So refractive medium, it brings the rays, colored rays of the light onto the retina. Okay, number one. So it is a refractive. How much refraction of the around? No, that's not an amount. 35% of the eye refraction is done by the lens. Okay. Then secondly, it has one more important function is that it absorbs the UV light. It absorbs the UV light because sunlight is the UV light, and once it absorbs the UV light, it does not allow it to fall onto the retina. And once it does not fall on the retina, that means the retinal damage is less. That's the nature has created light. Third important function of the human lens is the accommodation. Is it accommodation? So this lens can focus distant as well as near objects. It can change its refractive power. It can change its refractive power. So this is the function of the lens. So we will go something about the development of the lens. From which lens is developed? Yes, very good. It is developed from the surface ectoderm. It is developed from the surface ectoderm. So surface ectoderm, once if we have around 1 month or 7 days, there is a human brain. There is a human brain, what's called frozen Teflon. In that region, there is a surface ectoderm. There is a surface ectoderm. There is opposite thickening. That thickening is called lens plateau. That's called lens plateau. Once the lens plateau is formed, then it forms a pit. Once it forms a depression, what we call a pit, that pit that is detached from the surface ectoderm, it forms the lens vesicles. So that is a very important thing to understand. If somebody asks you what's the development of the lens, what should you tell? It develops from the surface ectoderm. So it does not develop from neural ectoderm. It develops from surface ectoderm. So the development part is developed from the surface ectoderm. Next year. Then if we go by the position in the eye, why is it located? That's called patternal fossa. What is patternal fossa? Patternal fossa is something is the vitreous. Here is the depression in the vitreous. There is a depression in the vitreous. That depression is called, that space is called patternal fossa. It is anteriorly limited by the eyes. So it is limited anteriorly by the eyes and posteriorly by the vitreous. So this is the eyes. So it is the eyes, this is the eyes, and this is the vitreous. This depression here is the patternal fossa. It is the vitreous. This depression is the vitreous, anterior base of the vitreous. So patternal fossa is the curve or this depression. So if we go by the dimensions of the lens, if we go by the dimension, what is the shape of the lens? Yes, it is biconvex. That is, anterior surface is convex, posterior surface is convex. Then what are the dimensions of the lens? How much is diameter? How much is thickness? Equatorial diameter of the lens is around 10 mm. How much? 10 mm. In children, it is less. At birth, it's around 6.5. What is the thickness of the lens? It's around 5 mm. You can remember like this: 10 is the diameter, 5 is the thickness. Half of that. Diameter is 10, thickness is 5. Finished. Then, if we think, one more thing is that if we think that what's the weight? What's the weight of the lens? In children, it is 100 milligram or around 90 milligram, but in adults, it is almost 3 times than this, 250 milligram. How much? 250 milligram in the adults. What is the differential power of the lens? People say around 17. Some people say 20. So 16 to 19 will be the range. Okay? So, but you can remember around 17 or 18. So, these were the dimensions of the lens. So, now we have a lens which is placed in the parallel fossa, which is 10 mm in diameter, 4, 5 mm thickness in 250 milligram adults. So, if we see what are its features which make it unique, first is what that student told, which is avascular. It is avascular. Once it's avascular, why did it get unique? So, it will get from the anterior chamber, from aqueous. The source of its nutrition is aqueous. It does not have any vessels. Does it have any vessels?
Embryology.
In the development, it has hyaloid group of vessels. Hyaloid group of vessels. But that is then
Regressed.
then that is obliterated. Then that is obliterated. But there is a canal in the vitreous, what is called a hyaloid canal, which is streak-like in the center of the vitreous. But that does not exist in the adults. So in adults it is avascular. That's to be understood. So once the avascular structure, so it needs a nutrition. Nutrition comes from the aqueous itself. Then we go to the structural parameters of the lens. What are the structural parameters of the lens? So lens has an epithelium, it has an anterior capsule, it has a posterior capsule. It has anterior capsule, posterior capsule. And that means it has capsule around it. Then it has a cortex, then it has a nucleus. Okay, nucleus, cortex, and the capsule. So we go first with the capsule. We go with the capsule. Lens capsule. First, can you tell me what are the attachments of the lens? Yes. One attachment is the granules. One attachment is the granules, which are attached to what? To the CVD body. They are arranged in 360 degrees. They are arranged in 360 degrees. Other attachments is what? Vigorous liquid. Wait. That is around the posterior, in the ring-shaped fashion. The posterior quarter of the lens is attached to the vitreous. What's called a hyaloid capsular ligament, or the other name for that is vigorous. And there is space between the posterior lens capsule and the vitreous. What is that space called? Retroventricular space, or other name for that is Berger's. B-E-R-G-E-R, Berger's space. So this is the location and these are the attachments of the human lens. Now we directly go to the capsule. Before we understand the capsule.
It's a capsule lens.
I want to make you understand one thing that.
चैलेंज सर आप सर एरो वाला है एक चल रहे सर चल सर पॉइंटर नहीं काम कर रहे सर चल रहे आप वहाँ से देखिए सर पॉइंटर नहीं खाली चल रहे सर कुछ पता आंसर ये वाली सर ये वाली
This is a very important diagram. So anterior, that's the anterior part. That's the anterior part. That's in front. And this is the posterior. This is the posterior part. So you have to understand first that anterior part has epithelium. Anterior part is the epithelium. Posterior part does not have epithelium. Okay, remember it for life. Anterior lens capsule has epithelium. Posterior lens capsule does not have epithelium. Where the epithelium is gone? What? Yes. So once basically the human lens has anterior epithelium, has a posterior epithelium. Suppose I draw it here like this. Suppose this is a human lens. So this epithelium was like this here. This posterior. This is posterior, and this is the anterior, like this. This has elongated. This has elongated like this, like this, like this, like this, like this, like this, and it has become an embryonic nucleus. It has become an embryonic. And these lens fibers, these epithelium, which was posterior, once it has gone in the center of this, it has become embryonic nucleus, and it has lost its organ, like mitochondria, nucleus, like that. So it forms the embryonic nucleus. It forms the embryonic nucleus. So in the adult lens, there is no posterior epithelium. Remember. So when the anterior epithelium has it's a sort of a cuboidal. It's a sort of a simple cuboidal, not only simple cuboidal epithelium. Then it has different regions. It has different regions. Suppose we go and these epithelial cells, once they reach here, like here, in this region, like this zone is called germinative zone. This is called germinative zone. So they get detached, they become polymer. Then they pass the equator. Then once they pass the equator, then they go posterior from the lens fibers. And this portion, this point is called bow region. Bow, B-O-W. That's called germinative zone, and this is called the bow region. Why it is called bow? Because here the differentiation occurs. The cell becomes elongated and it forms a lens fiber. It forms the lens fiber. Once it forms the lens fiber, the nuclei come anterior. The nuclei come anterior as they are going forward. So this is called the bow region. So it is basically the anterior epithelium in three parts. One is the cuboidal, then in the germinative they become polymer, then they get detached, they pass the equator of the lens, and then they go into the bow region where they get elongated and they form the lens fibers. Next. So this is what I told you about. Next. Lens fibers I told you. Okay. The lens fibers, they have some interdigitation between them. Just for the name, remember, there are two types of digitation. One is called ball and socket, and another is called downline grooves. They are interdigitation between the lens fibers. Next. So these are the different nuclei. They are the different nuclei. The center one is the earliest. And as we move out, it becomes the oldest. Okay. So if we go to the center, this is the embryonic nucleus. This is the embryonic nucleus. Embryonic nucleus means what? Which is present at 3 months. Which is present at 3 months. Then we have the outside this embryonic nucleus. We have this is called fetal. This is called fetal. That is from 3 months to 8 months or 9 months. This is called fetal. And it has sutures. And it has sutures. Because once this embryo is formed, this is formed, then the lens fibers form around it. Okay? And they form this fetal nucleus. They form this fetal and sutures are doing this. Lens sutures are doing this. How many sutures does lens has? No, how many? It has 2 sutures. One is the anterior and other is the posterior. So anterior is Y shape and posterior is inverted Y. So if we go like this, we form the fibers like this. Then the anterior becomes anterior Y like this. Okay, like this. So it moves like this Y. So anterior is Y, posterior is inverted Y. What is significance? You can develop sutural character. You can develop it. So if it is like upright Y, it's anterior suture. If it is inverted Y, posterior it is posterior suture. So this was the fetal. This was the fetal nucleus. Then outside the fetal nucleus, we have the third nucleus, this side. So what is this type called? It's called infantile nucleus. It's called infantile. Infantile is from birth or 9 months of intrauterine life to the puberty. And then outside this is the adult nucleus. Adult nucleus is then the adults, like beyond puberty or the adult. Outside that, what is this? There is the cortex. This is the cortex. This is the cortex, then this is the key or calcium. So, again I will remind you of these 4 nuclei. How many? 4 nuclei. One is embryonic, fetal, infantile, and adult. Embryonic is primary lens fibers. They are called what I told you. This epithelium elongates and it forms the primary. So no confusion there. Embryonic is the primary lens fibers in which the posterior epithelium is used. Posterior lens epithelium is used. That does not remain there. So there is no epithelium in the adult lens. So embryonic nucleus, that first 3 months, then from 3rd to 8th month it becomes infantile, fetal. It becomes fetal. So embryonic, then fetal from 3 months to 8 months. Fetal is the lens fiber which has sutures. And sutures for anterior and posterior, anterior upright Y, posterior inverted Y. Then outside that there is the infantile nucleus, which is from the birth to the puberty. Then beyond puberty, there is one more nucleus, that is adult nucleus. Because the lens fibers are continuously being formed, and once the lens fibers are continuously being formed, they come across the central cord. Okay, so they form embryonic, fetal, infantile, and adult. Again, embryonic 3 months, fetal 8 months, beyond 8 months, puberty, infantile, then adult. These are the 4. The capsule I have told, and this sutural cataract I told you how it is formed. Next. So what forms the embryonic nucleus? That's called primary lens fibers. Outside that, everything is secondary lens fibers. Okay. Especially the fetal nucleus. That is secondary lens fibers. Only the embryonic nucleus is primary lens fibers. Next. So this is the structure of the lens fibers in the fetal nucleus. In the fetal nucleus, in which there is the anterior Y and posterior inverted Y. This is the pattern of lens fibers. Suppose you can see here, this is the Y structure. main nucleus, which is formed by the continuously by the growing lens fibers from the germinating epithelium. Next. Okay, this is not that important for you. Just like forgot to tell you the epithelium which is present in the anterior capsule. That secretes the basal membrane. That secretes the basal membrane. Anterior capsule as well as the posterior capsule. What is the thickness of anterior capsule? What is the thickness of posterior capsule? Anterior capsule is thicker than the posterior capsule. Anterior capsule is around 14 microns. How much? 14 microns. And posterior capsule, in the region of posterior capsule, is 4 microns. 14 and 4. So it is thinnest. It is thinnest. And the periphery of the capsule towards the equator on anterior or posterior side is thicker. That's around 21 or 22 microns. So because posterior capsule is very thin, so this is why we can have posterior capsule ramp during surgery. Because there is only 4 microns. So once there is a ramp in posterior capsule during surgery, what will get? The width will come. Okay, that's the complication which can develop. So one more thing I want to tell about the applied anatomy of the anterior capsule or posterior capsule is that you can develop anterior capsular opacity. Anterior capsular opacity. In what condition? You can have metaplasia of the epithelium. You can have metaplasia of the anterior epithelium in the region of the anterior core. You understand my point or not? Suppose this is the anterior capsule. This is the anterior capsule. You can develop here cataract. This is the posterior capsule. Huh? So you can develop here. What can happen now? There is metaplasia of the epithelium. And in which condition is this? Second condition, atopic dermatitis. Atopic dermatitis. Okay? And there is one more. What is that called? I think that's called shield cataract. Shield cataract.
Radiation.
Huh?
Radiation, sir. Radiation की वजह से भी. Metaplasia because of radiation, sir.
Yes, metaplasia due to radiation. Metaplasia due to radiation. So you can have due to atopic dermatitis or due to radiation also. I think there is some term called shield cataract. Probably that is due to either radiation or due to atopic dermatitis. But that forms an opacity here near the anterior core. So once this capsule goes, this epithelium goes, form new lens fibers, it can form posterior capsular opacity also. It can form
Posterior capsule.
where? It can form here in the visual axis here. What's called? Posterior subcapsular cataract. That's called posterior subcapsular cataract. Why that form? Because there occurs dysplasia of the epithelium. There occurs dysplasia once this. Because I told you this is the germinated zone and this is the bowing. So here the dysplasia also. And once it occurs dysplasia, it forms the posterior subcapsular cataract. Once it forms an opacity here, what is this posterior subcapsular cataract is called what? Collically. It is called bread and butter appearance. What's called? Bread and butter appearance. Bread and butter appearance. So one more cause of posterior capsular opacity is what? Once we do what? We remove the lens, we do a cataract surgery, we put an intraocular lens. After 6 months, patient comes with a posterior capsular opacity. What's called PCO? What's called? PCO, postal capsule opacity. What's the cause of that postal capsule? Migration. Yes, very good. Migration of the cell. Because we leave some part of the equator of the anterior capsule, and those cells migrate to the posterior capsule. Okay, they migrate. So how we can avoid it? Central axis formation. Yes, central circular axis formation, which should cover the optic of 360 degrees. Number one, because that will not help. Number two, we try to remove the epithelial cell. How we can remove the epithelial cell? No, that's not the PCO has happened. They do the laser. Prevention, prophylaxis of PCO. How we on table we do that.
Irrigation
Yes,
aspiration.
Good. So that the epithelial cells come out. Or we can polish that. We can polish that so that the epithelial cells are shattered. So this is how we make the PCO occurs. So I told you about the epithelium, anterior epithelium, posterior epithelium, cataract, PCO, posterior rank. Then I showed you about future cataract. Then we have one more phenomenon, what's called clinically or surgically, human lens. Oh, madam, आप क्या कर रहे हो? आप आगे So clinically, surgically, into three parts. What's called first is the endonucleus. First is the endonucleus. Then is the epinucleus. Then is the epinucleus. Third is the capsule. So endonucleus, epinucleus, and the capsule. Endonucleus. We have epinucleus and we have the capsule. So once we do the CCC, then we try to put a jet of waves below the capsule. That forms a hydrodissection. That separates the whole nucleus. Nucleus plus epinucleus from the capsule. Okay, because nucleus will start rotating. Okay, the corticocapsular adhesions are broken. Then there is one more procedure that's called hydrodelineation. That's called, last time I had shown to some students. The hydrodelineation we put the cannula within the capsule, within the cortex, and the nucleus is separated from the epinucleus. That gives a golden ring sign. So that's called hydrodelineation. So that is the applied application of lens anatomy. So in hydrodissection, we separate both epinucleus and endonucleus from the capsule. And in hydrodelineation, we separate the epinucleus from the endonucleus. Okay. Next. PQ. Silence over, Mr. So on the slit lamp, we have some features of lens. What are the features of the slit lamp?
सही वाला.
This is the slit lamp picture of the nucleus. So anteriorly we have the anterior capsule. Okay, then we have the superficial cortex. Then we have the superficial cortex. Superficial cortex is divided into two. That's called C1 and C2. C1 and C2. Suppose this is the cornea, this is the cornea. This is the anterior capsule, and this is the C1 and this is the C2. That means C1, C2 means what? Superficial cortex. And C3, C4 is the deep cortex. So this is the C1A and C1B superficial cortex, C2 and C3 or the deep cortex. Then we have the nucleus. Then we have again the cortex. Then we have the posterior. So once we see in the slit lamp, we form an optical section. What's called optical section? It forms a narrow beam of light which goes through the cornea across the lens, and we see the layers of the lens. This is called optical section. Next, this thing I told you: nucleus, epinuclear plate, the endonucleus, and this. Next, and this is important. Then we have ciliary zonules. We have the ciliary zonules. What are the function of zonules? They hold the nucleus from the equator, all around 360 degrees. Before we go to these ciliary zonules, I forgot to tell you, the lens is biconvex. What is it? Lens is biconvex. Which surface is more convex? Posterior. The posterior surface is more convex, anterior is less convex. How you correlate radius of curvature with the convexity of the surface? It is inverse. So if the radius of curvature is less, it is more convex. If it is more, so anterior curvature is around 10 micrometers.
More.
How much? 10 micrometers. And posterior curvature is 6 micrometers. How much? 6. So posterior curvature is more curved.
So less good.
Now I come to the zonular part. The zonules are, they have the nucleus from the equator. So they originate from two places. Both are parts of the ciliary body. Both are parts of the ciliary. Basically, ciliary body is divided. Ciliary body is in 5 mm zone. How much? From the limbus. Anterior 2 mm is called pars plicatum, and posterior 3 mm is called pars plana. So they arise from pars plana and they arise from pars plicatum. So what arises from pars plana? Because that's plane. Why that's called plana? Because that's plane. What is called plicatum? Because this has
Projections.
valleys and depressions, and it secretes the aqueous humor. So what comes from the pars plana, posterior part, is called pars orbicularis. That's called pars orbicularis. What comes from the anterior, this pars plicatum, that's called zonular. That's called zonular. Zonular fibers. Those are called pars orbicularis fibers. So I'll just demonstrate you a bit. Just keep your mind on this lens. Suppose this is pars plicatum and pars plana. So suppose this is the lens. This is the lens like this. The fibers come from the pars orbicularis attached to the anterior surface in front of the equator. And from the pars plicatum, two fibers come. They attach to the equator and they go to the posterior. So it becomes a limb line. Okay. But anterior to the equator, they are 1.5 mm here. How much? 1.5 mm. And posterior is only 1.25 mm. So again I have divided fibers from pars plana go to the anterior. They form the anterior limb. Fibers from pars plicatum, they go to the equator and they go to the posterior. Okay, so there are 3 limbs. How many? There are 3 limbs like this. This is the anterior limb, which is around 1.5 mm in front of the equator. This is the posterior limb, which is 1.25 mm posterior to the equator. And this is the equatorial limb. This is the equatorial limb. And the base of this, because it's called the equatorial, that's called the zonular fold. That's called zonular fold. So it has 3 limbs. Anterior limb, which is formed by the pars orbicularis, pars plana, and the equatorial, and posterior, it forms a crisscross. You understand? So once it forms the crisscross, there are some spaces there. There are some spaces there. There are one more fibers which go to the hyaloid also, vitreous. That goes to the vitreous. Like this. Like this, if something goes like vitreous, there is space created here. That space is called space of Petit. That's called space of Petit. And there is one more space which is between the anterior limb and posterior limb. This is the anterior limb and this is the posterior limb. That's called space of Monover. That's called space of Monover. So space of Monover is between the anterior and posterior limb, and behind the posterior limb and between the vitreous fibers there is a space of Petit. So what is the importance of these zonal fibers? Yes. So there is accommodation. There is an accommodation. What is accommodation? Because lens has a unique property. You can see distance, you can see near. Once you see the near, the refractive power of the lens increases. What increases? Anterior or posterior? Anterior. So it becomes more spherical. So it's suppose it's like this, it becomes like this, and it forms a near image also on the retina. Once we are like 40s and 50s, then we need the glass for accommodation. Okay. So your accommodation goes. Your accommodation goes. Why does it go?
Weak service.
Yes, because of the ciliary body muscle is weakened or atrophied. How does this accommodation happen? Anybody can explain? I will tell you briefly, but you understand. Normally, ciliary body is posterior. Ciliary body is posterior, and the zonal fibers are tight. Okay, like this, like this. There's less convex lens. The fibers are tight. Once this ciliary body contracts, it comes forward. It comes sliding forward. Once it comes forward, the fibers, the zonules become loose. They become loose. Once they become loose, capsule distance. And once capsule distance, it becomes more convex. Okay, this is called accommodation. You understood or not? So I will again demonstrate. Suppose these are the so this is the ciliary body in relaxation. This is ciliary body in relaxation. Once it is in relaxation, lens is thin. Okay, and zonules are tight. Okay, once it contracts, this accommodation, they become loose. They become loose. The nucleus becomes big. It becomes more spherical. More anterior capsule becomes more spherical. Posterior capsule also becomes more, but by less extent. Anterior becomes more. And once it becomes more spherical, it increases its refractive power. Once it increases its refractive power, the near objects are also focused on the retina. So there are three things. One is ciliary body contraction. Other are zonules loosened. Third is the lens becoming more convex. What are the three important things regarding the accommodation? One is this. Other is what?
Meiosis.
Yes. Very good. Other is pupillary constriction. So one is the increase in the refractive power of the lens, mechanism I told you. Number second is the pupillary constriction, right? Third is the convergence. So these three things: increase in the refractive power of the lens, convergence of the eye, and pupillary constriction. So these are the three accommodation parameters. So once you go above 40, you lose this accommodation and you need glass for the near, what's called presbyopia. Other use of zonules, other clinical activity, zonules can become weak. weak, and you can have ectopia lens. You can have what's ectopia lens? The lens is not in its normal position. Okay, what is this fibers formed of? They are formed of fibrillin. They are formed of fibrillin. They are formed of fibrillin. So in this condition, this fibrillin becomes brittle. Yes. So clinically we have two conditions in which it becomes weak. One is marfans and other is homocystinuria. Homocystinuria. Marfans and homocystinuria. What is the difference between marfans and homocystinuria and ectopia? Remember again. Marfans is up. Marfans is up. Homocystinuria is down. Okay. Up and temporal. Up and homocystinuria is down and nasal. This is the difference. Marfans is up and temporal. Homocystinuria is down and nasal. Clinically, this is the difference. Next.
Sakya. चला किया मैंने.
Next. So, physiology of lens. Lens is very important. Lens is made up of two things more, 99%. It is simple. One is water, and other is proteins. After some more. So 66% water and 33% is the protein. That is around 99%. So it is in a state of dehydration. Because dehydration is needed, because it has to be transparent. So it should pass the rays. Then other minor constituents contain carbohydrates, other minor constituents contain lipids. Then we can have glutathione, then we can have phosphates, then we have vitamin C, which are important in the lens metabolism. Yes. This I told you. Next. So the lens proteins are generally three types. They are alpha, beta, and gamma. Alpha, beta, and gamma. They are called soluble proteins. Once these soluble proteins go into insoluble, the cataracts start. Okay. What are the insoluble proteins? Normally, the most important is the alpha crystallins. Most important are the alpha crystallins. If alpha crystals decrease, then if the insoluble proteins increase, it leads to the cataract formation. What are insoluble proteins? Next. Insoluble proteins are, they have own disulfide bonds. They have own disulfide bonds. And number two is there's one more protein, what's called major, what's MIP here? That's called aquaporin O. Aquaporin O.
It's an intrinsic protein. Intrinsic protein.
Yes. Major intrinsic protein. Other name for it, that is aquaporin O. It's same. Major intrinsic protein or aquaporin O. If its concentration increases along with the disulfide and superadded, if there is reduction in the oxidative enzymes. What are the oxidative enzymes? Glutathione. What type of glutathione?
Reduced.
Reduced. Yes. You are right. Reduced glutathione. Reduced glutathione. If there is decrease in the reduced glutathione and there is increase in the insoluble proteins, that leads to the cataract formation. And what are the other enzymes? Number one is reduced glutathione. What are the other two enzymes? catalase or superoxide dismutase. Catalase and superoxide dismutase, which are the antioxidative properties. Which are antioxidative properties. What has the oxidative property?
Free radicals.
Oxygen free radicals. Oxygen free radicals. Even you have glutathione disulfide, which is in the oxidative state, which is in the so that also causes the cataract. So these are the few things which lead to cataract formation. Then the lenticular metabolism. This is very important because lens is avascular. It does not have an oxygen supply. So main source of energy is glucose. Main source of energy is glucose. Superadded which pathway? Because there is no oxygen, so it will be an anaerobic glycolysis. Anaerobic glycolysis. So anaerobic glycolysis we have 3-4 things. We have 3-4 cycles. Next, we have glycolysis. That's the majority, 80%. Then we have the Krebs cycle, which is the minority, 5%. Then we have the hexose monophosphate state, that is the 15%. Then one more is the sorbitol pathway. One more is the sorbitol pathway. This sorbitol pathway is very important. This sorbitol pathway is very important. Next. Keep this here only.
Okay, sir.
So, if we, from our biochemistry, we know once glucose goes into glucose 6-phosphate, which is the enzyme? Hexokinase. It's a hexokinase. So, once glucose 6-phosphate goes to pyruvic acid, so it becomes a glycolysis. So, the generation of ATP is how much? 2. Okay? Then, if it goes to Krebs cycle, pyruvate goes into Krebs cycle, it forms carbon dioxide and water. So the generation of ATP is 36. How much? 36. Once this 36 ATP and 2 ATP, if we go then to this pentose pathway, what happens in the pentose pathway? Glucose 6-phosphate goes into what? Phosphoglutamate. And then the pentose, and it forms NADPH like that, from which the ATPs are generated. So it is a minor. It does not produce much ATP. Then we have the sorbitol. Then we have the sorbitol pathway. This sorbitol pathway is formed by what? It is formed by the enzyme aldose reductase. By aldose reductase. So it forms the sorbitol, then sorbitol gets transformed into fructose also. Okay? So this is the lens metabolism. So predominant metabolism is anaerobic glycolysis. So that means what? The pyruvate will form more in the lens. Okay. Then here I want to tell you this hexokinase is a very tricky enzyme. It's a rate dependent. It's a rate dependent. It's a limiting factor. So if you have more glucose in the eye or more glucose in the body, it will not go into glycolysis. So it will go into sorbitol. It will go into sorbitol, like in diabetes. And sorbitol leads to what? The cataract in diabetes. One of the postulated hypotheses is this sorbitol leads to cataract. Why does sorbitol lead to cataract? You get it? Yes, very good. Osmotic pressure. Now this sorbitol is within the lens, it will pull the water around it and it will change the refractive index of the lens and it will lead to, because I told you it's a dehydrated structure. Why does sorbitol become? Next question. There are so many other these pyruvates that anybody can tell me? Because the lens epithelium is impermeable to sorbitol. It is not allowed to leave. You understand? Sorbitol gets accumulated because the lens epithelium is impermeable. Okay? So once there is more glucose in the aqueous, it will lead to more glucose in the lens. Whereas if more glucose in the lens, it will lead to the limitation of the hydrokinase. Once it will lead to limitation of the hydrokinase, it will lead to more sorbitol formation. More sorbitol formation, it gets trapped in the lens. It does not leave the lens because lens capsule is impermeable and it leads to the osmotic gradient. What else can lead to osmotic gradient? If there is high salt in the body. Okay, once there is more sodium in the body, more sodium in the aqueous, more chloride in the aqueous, that will go into the lens, that will cause more osmotic barrier. What else can cause? There are some things like lens proteins which degenerate. The way I have told, the alpha crystals come down. Okay, the insoluble proteins increase. This happens in nuclear sclerosis. It happens in nuclear sclerosis. And the nuclear sclerosis, what happens? The refractive index of the nucleus increases. Okay, and eye becomes myopic. Eye becomes myopic. What's called a second sight. What's called second sight. They are not able to see distance, but near they have very good vision because of the myopia induced by the high refractive index. So the old age persons or people from the village are telling the doctor, our father has very good vision. He has a very good iman. He has a very good iman. He has got a very good near vision. But they don't know he has cataract. He got the vision because of cataract. Because after 40, 50, you don't have near vision. But now he has to regain the near vision. And they say he is very good. Even at 90, 100, he has a very good vision. Because his near vision is increased. Do you understand? Suppose in diabetes, one more situation I tell you. I want to clear this to you. Suppose in diabetes, now sorbitol goes, is trapped in the lens, and more fluid goes into this. What will happen? It will increase the refractive index. It will increase again the eye will go into high myopia. Okay? So hyperglycemia causes what?
Myopic shift.
Myopia. Hyperglycemia causes myopia. Remember, because the osmotic pressure of the lens is increased. Osmotic pressure of the lens is increased because more fluid goes into it. Because high glucose in the lens and that attracts more water, and that is the refractive index of the lens increases and the eye becomes myopic, like the nuclear sclerosis. But here I want to again clear. If there is hyperglycemia, it will also cause cataract. If there is hypoglycemia, it will also cataract. So hyperglycemia, I told, because of the osmotic effect, it will lead to cataract. What about the hypoglycemia? Because it will cause a nutritional deficiency. It will cause it because the source of lens metabolism, lens energy is what?
Glucose.
That's glucose. Once that is depleted, it leads to its degeneration or to the cataract formation or to the opacity. So, last line of two words. So, look at this slide. This is a very important slide. This is a very important slide. Oh, Mr. You come forward. Come forward. So, look at this slide. Look at this slide. Basically, the anterior capsular epithelium, it has a sodium potassium pump. It is a sodium potassium pump. Posterior capsule does not have any pump. So once there is a pump mechanism in the anterior capsule or anterior epithelium, what will happen? Sodium will come out from lens to the outside. In the aqueous, sodium will be high. Okay. And potassium will be intracellular within the lens wall. Okay. So potassium is high within the lens, and sodium is high aqueous. This is due to the sodium potassium ATPase bond. Okay. So one of the features, one of the important things is potassium is high in the lens and sodium is low. Because if sodium gets high, it will drag the water. And once it will drag the water, it will need to get that. So nature has created it like that. Sodium is pushed out, sodium is pushed out of the lens, and potassium goes in. Then you have like the sodium, the chloride is also less. Once sodium will come to the aqueous, chloride will also flow in. Okay. Then glucose is metabolized in the lens. Glucose is metabolized, so in the lens it will be less. So aqueous will be more. Then you have the proteins and amino acids. Because lens fibers are proteins, they are amino acids. So they are very high. Okay. So amino acids are very high, like 33%. Or proteins are very high, 33%. Same is about the filament fluid. Reduced fluid is within the lens, so its concentration is very high in the lens. So these four are types of lens. One proteins, other the glutathione, 12. Then you have the potassium, and lactic acid. Okay, lactic acid is 4. Amino acids. So one is amino acid. Forget about lactic acid. One is amino acid, other is the protein. Because lens is protein, so both amino acid and both proteins will be this. Okay. Then you have potassium. Then you have the potassium. Then you have the glutathione. These four. Remember these. Amino acids, proteins, then the potassium and the glutathione. Sodium chloride is up, water is out, okay, and the glucose is also out. Because glucose is metabolized in the lens, how it can be high in the lens? And that too by anaerobic glucose. So you need more ATP. So it will rapidly this metabolize. There is one leak through. There is one leak through. What is this leak through? Last slide, that's all. I will explain it, that's all. There is sugar tobacco, otherwise I will displace everyone. So there is this anterior capsule and this is a posterior capsule. The sodium moves out and potassium moves in. So you know from sodium, potassium come. 3 sodium will come out, 2 potassium will go in. Okay. This potassium will go in, it is here from the posterior capsule into the pupils. Okay. But this is passive. Here it is active, here it is passive. And this sodium comes from the pupils to the lens. It comes to the lens and it goes down. This is called a leak. It's called leak. Sodium coming from the lens to the aqueous. And potassium from the aqueous to the lens to the pupils. This is called a leak. The anterior has active transport and posterior capsule has passive transport. Then we have some have you heard about the drugs? Have you heard about drugs? A combination I was telling you about, which system is responsible for a combination? Parasympathetic or myopathy. Which nerve is involved? Third cranial nerve, oculomotor. Which drugs cause this?